Welcome to this lecture on prokaryotic transcription regulation. Today, we are going to explore one of the most fascinating and elegant systems in biology. We will learn exactly how simple, single-celled bacteria make incredibly smart decisions about which genes to turn on and which to turn off, allowing them to survive in constantly changing environments. Before we dive into the molecular details, let us build some intuition. Imagine you own a massive factory that manufactures heavy winter coats. If it is the middle of a scorching hot summer, would you keep your machines running at full speed? Of course not. That would be a massive waste of electricity, raw materials, and money. You would only turn the machines on when the weather gets cold. Bacteria operate using this exact same brilliant logic. They have thousands of genes acting as instruction manuals, but they do not build all of them all the time. They regulate their transcription to save energy. Now that we understand why bacteria regulate their genes, let us look at how they actually do it. In prokaryotes, genes that are involved in the same task are often grouped together on the DNA into a single functional unit. We call this unit an operon. Think of an operon as a complete workstation. It consists of the structural genes themselves, which are the actual blueprints. Right next to them is a promoter region, which is the landing pad where the enzyme RNA polymerase binds to start reading the DNA. Finally, there is an operator region, which acts as a physical switch to control the whole process. Understanding the operator switch brings us to the proteins that actually flip it. The most common way bacteria turn off a gene is by using special proteins called repressors. These repressor proteins are designed to bind directly to the operator switch on the DNA. When a repressor is attached there, it acts like a giant roadblock. It physically blocks RNA polymerase from moving forward and reading the structural genes. By stopping transcription in its tracks, the repressor acts as a molecular brake, ensuring the cell does not waste energy producing proteins it does not currently need. While repressors act as brakes, the cell also needs a way to speed things up. This brings us to activator proteins, which do the exact opposite of repressors. Instead of blocking the path, activators bind to specific sites on the DNA to actively help RNA polymerase attach to the promoter much more efficiently. By stabilizing the enzyme, they significantly increase the overall rate of transcription. You can think of activators as molecular accelerators, stepping on the gas pedal when the cell urgently needs a large amount of a specific protein. Now that we understand both the brakes and the accelerators, let us look at why this matters in the real world. This dynamic system is absolutely fundamental to medicine. For example, infectious bacteria use these exact switches to survive inside the human body. They keep their disease-causing genes turned off to save energy, turning them on only when they detect the warm environment of a host. Furthermore, scientists have hijacked these natural bacterial switches for biotechnology. By placing the human insulin gene under the control of a bacterial operon switch, we can force vats of bacteria to mass-produce life-saving insulin for diabetic patients. To truly master how these biological switches work, we must look at the underlying chemistry. The probability that a gene is turned off depends on the fraction of operator sites bound by a repressor. We describe this using the fractional occupancy equation. Y equals the concentration of R divided by the sum of Kd and the concentration of R. Here, Y represents the fractional occupancy, which is the proportion of DNA switches that are blocked, ranging from 0 to 1. R is the concentration of the active repressor protein floating in the cell. Kd is the dissociation constant, which measures how tightly the repressor grips the DNA. If the repressor concentration increases, Y gets closer to 1, meaning the gene is heavily silenced. Now that we have our formula, let us see this with a concrete example. Imagine a bacterial cell where the dissociation constant for a specific repressor is 10 nanomolar. The concentration of active repressor in the cell is currently 40 nanomolar. We want to find out what fraction of the operons are repressed. We start by taking our fractional occupancy equation and substituting the given values. We replace the repressor concentration with 40 and the dissociation constant with 10. The numerator represents the repressor present, and the denominator represents the total binding potential. Simplifying this, we divide 40 by 50. This gives us a final fractional occupancy of 0.80. Physically, this means 80% of the operons are currently blocked. So we have calculated a fractional occupancy of 0.80. This brings us to an important biological insight. This result means that 80% of the operons in the bacterial population are currently bound by the repressor and turned off. But notice that it is not 100%. Physically, this tells us that the gene is highly repressed, but it is never completely silenced. It allows a tiny baseline level of transcription to occur. We call this baseline leakiness. It is actually a crucial survival feature, ensuring the bacteria always have a few enzymes ready, just in case the environment suddenly changes. Let us continue with the same bacterial cell to see what happens when the environment changes. Suddenly, the bacteria encounter a new food source, which acts as an inducer molecule. This inducer binds to the repressors and inactivates them, dropping the active repressor concentration down to just 2 nanomolar. The dissociation constant remains 10 nanomolar because the physical nature of the DNA interaction has not changed. We plug our new, lower repressor concentration into the equation. We get 2 divided by the sum of 10 and 2, which simplifies to 2 over 12. Calculating this gives us a new fractional occupancy of approximately 0.167. The math perfectly mirrors the biological reality of the repressor being neutralized. Now that we have calculated this new value, let us understand its impact on the cell. The calculation gave us a fractional occupancy of roughly 16.7%. This means that only about 17% of the operons are now blocked by a repressor, which is a massive drop from the previous 80%. Practically, this massive drop in repression means the physical brakes have been completely removed from the DNA. RNA polymerase is now free to attach and transcribe the genes needed to digest the new food source. The cell has successfully and rapidly adapted to its new environment. To solidify everything we have discussed, let us look at this diagram comparing the two states of the operon. In the top half, we see the repressed state. Notice the blue repressor protein sitting directly on the operator region of the DNA. Because it is parked there, the RNA polymerase is physically blocked, marked by the red cross, and cannot reach the structural genes. In the bottom half, we see the induced state. An inducer molecule has bound to the repressor, pulling it away from the DNA. With the roadblock gone, the RNA polymerase freely moves forward, indicated by the green arrow, transcribing the genes into messenger RNA. This visual perfectly captures the molecular switch in action. We have covered a lot of ground today, so let us summarize the most important ideas. First, Prokaryotic transcription regulation is an elegant system that allows bacteria to conserve energy by only making proteins when absolutely necessary. Second, they achieve this by grouping genes into operons, using repressor and activator proteins as physical molecular switches. Third, we saw how the fractional occupancy equation allows us to mathematically predict exactly how sensitive these switches are to environmental changes. Understanding this dynamic control mechanism is not just key to bacterial survival, it is the very foundation of modern genetic engineering. You now have a solid understanding of how gene regulation works at both a biological and mathematical level.